I'm now very pleased to introduce Professor Jeff Lichtman, Professor of Molecular and Cellular Biology at Harvard. Professor Lichtman is renowned for his work creating a process called Rainbow, like rainbow, a major contribution to the study of neural connections in the brain, in which individual neurons in the brain are flagged with distinctive colors. Please join me in welcoming Jeff Lichtman.
Thank you very much. It's my pleasure to be here. My main role, I think, is, and a really distinct pleasure, is to introduce my colleague and friend, Sebastian Seung, who is the author of this new book with this intriguing word, Connectome, as the title. I'll tell you a little bit about why I know Sebastian. He is trained as a physicist, and I am very much at the opposite end of the spectrum, at the wet, squishy end. I went to medical school, and I have also a PhD, and my interest is in brains, absolutely. And it is surprising that someone like me and someone like Sebastian would find common ground. In fact, he needs me, and I need him, I think. Would you say that true?
It's like marriage.
It's like marriage. But it's not really marriage. I want to make that clear here. I am married already. Let me tell you a little bit about one of the most daunting areas of ignorance humans have. And despite our attempts to understand the natural world in great detail, we have still made, I'm sure many of you will be surprised to hear this, almost no inroads into understanding ourselves and our brains. The other organ systems of the body, we've done great. We really understand lungs, livers, kidneys, secretory organs like glands. But when it comes to the brain, we still are in truly the infancy of understanding this organ. And a good example of this is that many of the most serious disorders of the brain, things like schizophrenia that afflict about 1% of humankind, learning disorders and autism spectrum disorders in childhood, have unambiguous behavioral manifestations that almost anyone can see just dealing with a person who has a problem like this for a short amount of time. But there is no histological trace whatsoever. There's nothing that we know that shows exactly what's wrong with these brains. And it's not that there aren't things wrong with them. It's just that in order to understand what's wrong, one has to appreciate that the brain is just not like any other organ system. It's made up of nerve cells called neurons, and they are interconnected in a vast wiring diagram that we do not understand, much less have even a copy of yet. And mapping this is a goal for the future. It's a goal that we're actively involved in now. And this book, I think, gives you a good sense of how the early people who are foolish enough to go down this path, and I include myself in this group, are operating, and how people like Sebastian are trying to make sense of the data that is generated. I'm a member of something called the Center for Brain Science at Harvard, and one of the missions of the Center for Brain Science is to push the envelope of understanding of the brain by going into fields where there isn't a great deal known. As I said, with the brain, this is not a big problem. Almost any direction you go in brain science, you very quickly get into areas of terra incognita where nothing is known, and nowhere is this more obvious than in the wiring diagram of the brain. Sebastian is a person who appreciates that if you ever want to understand a map of the brain, you're going to have to deal with data sets and methods of mapping connections that far exceed the talent base and window of knowledge of biologists. He's aware of the limitations of biology. We're very good at dissecting brains. We're even good at taking pictures of brains. But turning that information into something useful requires a kind of physicist's precision and a kind of computational comfort that most biologists don't have. So it's not a coincidence that Sebastian is not just a physicist, but also a computational neuroscientist, a neuroscientist who uses the power of computers in order to make sense of things that are beyond the ability of simple human brains to understand. One of the great paradoxes is that we're using our brains to try to understand how brains work, and this is already a big problem because the machine that underlies thought is much more complicated than the thoughts, at least that come out of my brain. And this is a great paradox about how we're ever going to understand ourselves. I don't want to really spend any more time talking. I want to introduce Sebastian so he can give you his sense of why a book like this was worth writing. And then afterwards, I think Sebastian will take questions, and there will be perhaps some questions that are a little more wet and squishy or related to disease, and I may chime in as well. But would you help me welcome Sebastian Seung?
I'm really grateful that you come here to share what's a very special occasion for me. I have worked very hard on this book, I assure you. I feel like a student assuring the teacher that I've worked so hard on the homework. But it's been an intense process for me. I've learned a lot by writing this book, and I want to share this with you. I've never written a book before. It's the first time, and this is the first event in the book launch. So you've come at a very special time in my life, and thank you all. Now, I've said a lot about how I feel, but actually, the reason we're all here is not me. I persuaded my friends to come, but that's not the reason. It's not about me, and it's not even about this book. All right, it's a book signing, but it's not really about this book. I would like to propose that we're here to celebrate an idea, a powerful idea whose time has come, and that is the idea of the connectome. All right, so this is not a new idea. It's been kind of maybe an unformed dream of neuroscientists for the past century, who have dreamed of understanding how neurons are connected to each other. And it's been a private obsession of a small group of neuroscientists, including Jeff and myself, who might be regarded by other people as a kind of cult. Or I don't know. We're a special group of people who believe that this is the right way to study the brain. And I have to tell you that that the brain, so Jeff talks about the brain as this object that's very difficult to understand. And if you were to see the discussions between neuroscientists, how we war with each other about what is the right way to understand the brain, it's like hearing philosophers talk. I mean, we have experiments, but when we talk about the future, there's a war of opinions. And Jeff and I belong to a group who believe that we have to look at the brain and see how it's connected. And that's going to be a major method of understanding. So this has been a private obsession of us. And this book launch, you're at a historic event because this is the coming out party. This is the coming out party of the Connectome. All right? And so that's why we're here today. And it's about an idea. It's more powerful than a particular person or two scientists or even the whole neuroscience community, because this is going to take the work of generations. So it's a quest, and it's a quest that includes all of us, because I think that the Connectome has a lot to influence about the way that we think of ourselves as humans, because our brains, our minds are ultimately what is behind our humanity. Okay? So let me say a few words then about what the book is about. I'm not going to actually, I'm going to summarize. I'm not going to make arguments. The book is one long argument, right, about why I believe these things, but I'm going to make some claims. And we can actually discuss them through interaction and question and answer. But here's the summary. So the first topic that I address in this book is, why should we study neurons? All right? Now, for most people, neuroscience is not about neurons. It's about brains and big pictures of spots on brains that you see in the newspapers, fMRI. That's really about brains and brain regions. But any single one of those brain regions that lights up in an fMRI study, a brain scanning study, it's got 100 million neurons in it, right? And what we're saying, what Jeff and I are saying, is that we've got to move beyond brain regions. We have to understand the brain at this level of neurons. And in fact, what I would say is real neuroscience ultimately is about neurons. We have to understand how the brain works, emerges out of this incredible assembly of these small elements. Now, that's a hard a hard claim to make because people say, well, it just seems so remote. These little itsy-bitsy neurons, 100 million of them, it just seems remote. How could I possibly, I mean, how could that be connected to my thoughts and my feelings? It just doesn't make any sense. But the first two chapters of this make the argument for why. Now, the second kind of claim, which is also covered in this first part, is that we have to understand how those neurons are organized. The secret to understanding the brain as an assembly of neurons is not to regard it as a sea of neurons, a bag of neurons, but to understand the intricate organization. And here, we really need images. Jeff is in the business of producing spectacular images of the brain, and you can go to the book website if you want to see those images. But since this is a Harvard bookstore, and this is this anachronistic thing called a book, we are relying on the power of words tonight. So I would like you to think about, we'll try to sort of describe it in pictures. But after you go home, look it up online, and you'll see the beautiful images. All right, but we have to look at neurons, and we have to see how they're organized. We have to see how they're connected with each other. And so that's the notion of the connectome. The connectome is the map of connections between 100 billion neurons inside your brain. You can imagine it like, let's say you're flying in an airplane, right? And these days they don't even give you good food on airplanes. You're already kind of unhappy. You're bored too. So you pick up the magazine and you look at the back of the magazine and you look at what's on the movies. They all look bad. And then you look at you're desperate for stimulation, so you look at the flight map, right? So there's all those cities with the 3-letter codes, and there's all those lines between the cities, which are the routes that fly between the cities. So think of each of those cities as a neuron, and each of those lines as a connection between two neurons. And then think of that map multiplied to 100 billion cities. And from every city, there's 10,000 connections to other cities, right? That is the organization of your brain. And it would never fit inside the back of that airline magazine. And that's the kind of thing that Jeff wants to analyze images of, take images of, and we want to extract the connectional organization of the brain. Okay, so that's the connectome. The connectome is the complete map of connections in your brain. Now, the second question, of course, is, why should we care? Why should we care about the connectome? Why should we care about connections between neurons? And that's what the next part of the book goes into. And the first reason is that experiences, it turns out that as you go about your life, your experiences excite patterns of electrical signaling in the brain, and those patterns leave traces on the connectome. They actually change your brain wiring. They change the way that your neurons are connected. And those changes are the way that you store memories. At least that's the theory of neuroscience. We've never been able to really test that because we haven't been able to see connectomes. But the theory goes that experiences get stored in your brain as memories through changes in your connectome. The next reason that we need to study connectomes is that everybody knows that genes matter. Genes have an effect on your brain and your mind. We know that from cases of mental disorders. Certain mental disorders are associated with genetic differences. And so why do genes, anybody could tell you genes have an effect on the brain. Okay, genes have an effect on the brain. They have an effect on everything. But how exactly do genes have an effect on the brain? If in order to move beyond philosophical controversies about nature versus nurture, what's genes, what's environment, we have to ask more specific questions. And one of them would be, how exactly do genes influence the brain? And a powerful way is by influencing the way, the whole process by which brains wire together during the development of the brain. And as a child, as a baby, your brain is wiring up, creating that connectome. And so an important mission of neuroscience is to discover how genes control that process. All right, so in a nutshell, the connectome is where nature meets nurture. And I would argue that the connectome is the basis of our personal identity, our memories, our predispositions for many mental disorders, our personality, our intellect. That's a hypothesis. But there's a lot of evidence to suggest that this is a plausible hypothesis, that our identities are somehow encoded in our connectomes. And that is a much more powerful kind of theory than saying, for example, that our identities are based on our genes, because our genes never change. Our connectomes can change, with difficulty, probably, right? So in short, if we ever are to have a science of personal change, we have to have a science of connectomes. We have to understand how connectomes change. All right, so I think that's the number one basis for why we need to understand connectomes. So then the next part of the book goes on to connectomics. So, you guys ask, well, if you guys are so smart, how come you haven't figured it out by now? It's been 100 years. Well, maybe we're not that smart.
That's true.
But I don't like to say we're not that smart because we're professors. We have to make a living. So I want to blame our technologies. I want to blame, it's a poor carpenter that blames his tools, but I want to blame our tools. If you don't have a microscope, you can't see bacteria. If you don't have a telescope, you can't see the heavens properly, right? So we have lacked the technologies in neuroscience to truly understand the brain as a collection of neurons. And so this part of the book is about those technologies and how they're going to change the way that neuroscience is happening, the way that neuroscience it's going to be a revolution in neuroscience. We're at a very special time in the history of neuroscience because of the new technologies. And some of these technologies are those for mapping out brain connections. And the final part of that, the final chapter of that part talks about the problem of developing new therapeutics, using our understanding of the brain to figure out how to change the brain, right? To help people recover from brain injuries, help people recover from mental disorders, to help people learn better, changing the brain. And this is a kind of important dream. It could seem a little bit scary to people, the idea that neuroscience might aid us in the quest to change ourselves. And this was brought up, of course, by people like, this kind of fears were brought up by people like Aldous Huxley back in the early 20th century in books like Brave New World, when they talked about the potential to use biotechnology to change humans and animals. And so I give my take on what the dangers might be. But then I go on and I take on people who love change, the transhumanists, the people who believe that neuroscience will give us the capability to change human nature in a new way. We won't have to wait for evolution to do that. We'll be able to really change humanity. And so one group of those people are the people who practice cryonics. They freeze themselves after they're dead with the goal of possibly resurrecting them. They hope that a future civilization will resurrect them, advanced civilization of the future. And the last chapter is about the people who want to do away with their bodies altogether. They want to take their brains, their minds, and transfer them to computers and live in this paradise of silicon as computer simulations, thereby achieving immortality. And these are fantastic ideas. They're science fiction. And of course, people have philosophical arguments over whether it's possible. My take on these ideas is that they do sound crazy, but we can actually think of ways of scientifically testing them using the idea of the connectome. Right? We can't prove they're possible, but we could think of ways to prove that these dreams are impossible through the connectome. And science is often about showing something as false. If you can't show something as false, it's philosophy. Okay? Right? Nobody can prove it's false. In science, you can't prove it's true, but you can sometimes prove things are false. Okay, so how am I doing on time? What time is it? 7:44? Oh, it's 24. All right, so I would like to move on to questions, but just I hope you'll indulge me if I just read a little bit from the book. Is that okay? All right. So I will read just two excerpts, and I will read actually from the final chapter on the people who want to live forever in a computer, because it's kind of fun. So the title of this chapter is Save As. It's distressing how little they tell us about heaven. We can at least imagine the gates. They are pearly and perched on a cloud. St. Peter stands guard, ready to make sinners sweat by posing tough questions. But what is it like inside the gates? Everyone wears white. I'm not sure how I feel about that. The harp is the only accessory, and angels abound. These snippets of information aren't much to go on. Only recently did I realize why religions might prefer to be vague. People would rather fantasize about their own heaven than have one thrust upon them. In the world's cultures and religions, conceptions of heaven have evolved slowly throughout history. Late in the second millennium, a radically new one emerged. Heaven is a really powerful computer. I don't mean that ecstatic look some nerds get when fondling their laptops. Let's not mistake such fetishism for a sign of spiritual enlightenment. But then again, why do these people spend so many of their waking hours online? Would it be too far-fetched to say that they thirst for transcendence, that they yearn to escape the inadequacies of this body and this world? While online, teenagers can forget the embarrassment of their pimply faces and underdeveloped physiques. People can take a pseudonym, alter their age, or masquerade with a photo of their dog. Netizens are free to be who they want to be, rather than who they really are. A body chained to a computer, glassy eyes staring at a glowing screen, and fingers pecking away on a keyboard. That's a slightly less corporeal existence, to be sure, but I would only call it purgatory. It's still not what I mean by the new idea of heaven. Some nerds want more. They would like to discard their bodies completely and transfer their minds to computers. The idea of living as a computer simulation has been embraced by science fiction, which calls it mind uploading, or uploading for short. It's not possible yet, but perhaps all we have to do is wait for computers to get more powerful. Video games are stunning proof that computers can simulate the physical world. Every year, the scenery looks more detailed and lush. Every year, bodies move in more lifelike ways. If computers can do that, why can't they simulate minds? It's no exaggeration to compare uploading with ascension to heaven. Just think about the word itself. Uploading gets the direction right. As most agree that heaven is located in a high place. Some devotees prefer to say mind downloading, but they are in the minority. It's not hard to understand why. Downloading sounds suspiciously like going to hell. All right, so that gives you a flavor of some of the pros. Why don't we take some questions? So to either me or Jeff. Yeah, so the question was, I made big claims about how this defines how the connectome, our connectome might be sort of the basis of our personal identity. And the question is whether that has been investigated in animals. And indeed, in the book, I lay out some possibilities for investigating this in animals. For example, trying to read out the memory of a bird's song from its brain. But that's never been done before. What has been done is studies of the worm connectome. That's the only connectome that's been completely mapped. And worm connectomes are much more similar to each other across individuals than ours. We have huge differences. And as Jeff likes to say, that's why he thinks that we're more unique as individuals than worms are. But even in worms, there are slight differences in the connections. And it hasn't been adequately investigated whether those differences are related to differences in the worm behavior. But a simple answer would be that we know that in humans, really big differences in connectivity do lead to differences in people's behavior, right? If you cut the big bundle of connections between the left brain and the right brain, it has some effect on what that person behaves like or can do. So we have indications like that. But whether the fine detailed connections of the brain actually encode your identity, we don't know because we haven't been able to trace those connections yet. What's new is we've never had technologies for investigating connections between neurons. So the idea that connections between brain regions are important has been, of course, part of neuropsychology since the 19th century. And we've had technologies for tracing those connections. But the detailed fine structure of connections between individual neurons, that is something that people could only dream of. All right, so the new technologies are the excitement.
Yeah, I mean, this is a very good point, that a structural map of a dynamic system, you're using a static picture to try to understand something that if we freeze your brain and look at it one moment in time, all the things that make a brain interesting have evaporated from it. One way to think about this is to say that whatever is different between a synapse that is strong or extra potentiated relative to another must have an underlying structural basis. It's just the structure you may be looking for here now is not so much who is connected to who, but how many synaptic vesicles are in a nerve terminal, or how big the contact is between one nerve cell and another. And all that is addressable. You can see all that. So in fact, one hopes to be able to look at that as well. The other point you mentioned, though, is that the brain is a chemical factory as well. There are all these neurotransmitters, and a structural map doesn't show you pictures of neurotransmitters. But there are enzymes that make these neurotransmitters that are in some cells and not others, and there are antibodies that can stain the sites where these chemicals are. So it is the proteome meets the connectome. You put the protein map on top of the connectional map, and between those two kinds of things, looking at the inside structures of synapses and the chemical makeup, one would have a pretty good idea of exactly what is going on, one thinks. I can say a little bit about the new technologies. I think most people, when you think of a microscope, you think of a human being sitting there and looking at something. And when you think of a telescope, you think of a human being peering through a little hole at the heavens. What's happened is people have come up with ways of industrializing the looking. Where the humans are not actually doing the looking anymore. The machines are doing the looking, and they're looking every day and every night, even Christmas Eve, New Year's Eve, even during the Super Bowl. They're just working away, it doesn't matter where they are, all the time generating information. So there are several approaches, but the most powerful ones probably are the ones that have enough resolution to basically see the entire wiring diagram. And those are ones that use electrons to do the imaging. These are electron microscopes, and they're often scanning electron microscopes, and they're looking at thin slivers of the brain, one at a time, but each sliver is on top of the next. So they take a picture of one sliver and then the one underneath. The thickness of these slivers are about 30 nanometers. That's 300 hydrogen atoms thick. It's about 10,000 times or maybe 1,000,000 times thinner than a human hair. I mean, incredibly thin sections. But if you look at a section and then you look at the next section and the next section and play these images as a movie, you see these wires just as if you're slicing through spaghetti. And you can just trace them, especially with tools like the ones that Sebastian makes. Yeah, we're talking, it's like a house, that range, each machine. But the amount of data you have to acquire, just to give you a rough idea, a cubic millimeter of brain, which is the size of a voxel of an fMRI image of blood flow in the brain, these very common tools that you see pictures of in magazines all the time. One of those voxels, 1 cubic millimeter of brain, in terms of image space, has about 1,000 terabytes of images. Terabyte is 1,000 gigabytes, so it's about 1,000,000 gigabytes per cubic millimeter, which is 1 petabyte. So this is...
it's like a billion images from your digital camera.
Yeah, 1 billion images from your digital camera is each cubic millimeter of brain. A human brain, if you got the images of the whole brain, equals the digital content of the world right now.
So the question is, how long will it take? That's always the, I'm saying this is the beginning. Jeff and I believe this is the beginning of a revolution. But the question is, well, will the revolution be over soon, or how long will this take, right? So revolution maybe is not the right word, because I guess the best comparison would be, in a favorable case, to the computer revolution. The computer revolution has been increasing for decades. the power of computers. And we hope that connectomes will be like that, but it could take decades to get an entire human connectome at neuronal resolution. And in fact, what we're happy with right now is a cubic millimeter, just a small chunk of human or mouse brain, but even that is going to tell us a lot. So we can do science as we scale up. Well, there's two points, really. Actually, the slogan of the book is, "You are more than your genes, you are your connectome." And so the obvious practical application would be in understanding differences between people. So why are we different, right? And people often say, oh, you know, Johnny's just that way. His brain is wired differently. But in fact, we don't have the technologies. We haven't had the technologies to really investigate that claim in a scientific way. If we could find something that's truly distinctive of a brain of somebody who has autism or schizophrenia, that would be a step towards a scientific development of treatments for those maladies, right? So it's actually something that people don't realize, right? If everybody now accepts the idea, oh, autism, schizophrenia, brain disorders. But actually, if you look at the brains of people with autism and schizophrenia after death, let's say with Alzheimer's, you can look at someone's brain after death and see clearly with a microscope they had Alzheimer's. Autism and schizophrenia, the brain looks normal. Right? Or there are abnormalities which are not consistent. So, you know, everybody has brain abnormalities as they get older, different ways, but there's not something that's consistently there for autism and schizophrenia. So we have a lot of indirect evidence they're brain disorders, but no real smoking gun proof. And I would say that the study of mental disorders is like studying infectious diseases before you could see bacteria in a microscope. You can't see what's wrong. Well, the obvious analogy would be that, so let's say we have the genome and the connectome as analogous, right? So I guess the question was, well, we have the genome, but it hasn't solved every problem in biology yet. It hasn't solved every problem in medicine, right? Well, isn't it the same case for the connectome? Even if you had the connectome, would something be missing? That's the question. So neural activity would be an analog. So genes are expressed into RNA and proteins. In order to have effects, genes get expressed into RNA and proteins. In the same way, the connectome inside my brain has no effect on brain function unless it expresses activity patterns. Okay, so it's the patterns of signals inside the brain. So we have to understand the relationship between neural activity and connections. So the connectome alone is not enough, right? But and in fact, we have to understand all levels in the brain: genes, connections, and activity, all of them. But I emphasize the connectome in this book because really, I think that's the fundamental quantity behind personal change. Because activity changes all the time. I quit smoking every day, right? That's neural activity. But quitting smoking for a lifetime, that's a change in the connectome. At least that's the hypothesis. Okay?
Yeah, I mean, one way to think about this is that for most machines, you think the structure of the machine explains its function. The brain is a unique organ in that the structure explains the function, but then the use of the machine, the experience, changes the structure. So you have this loop. Loops are horrible because there's no cause and effect anymore. Everything is affecting everything else. Humans hate to think about loops because you don't know where to start. Everything is affected by everything else. But that is the conundrum, and we hope that the way in which experience changes the brain is not an infinite number of ways, but that there are some rules to those changes, that we will see structural motifs, if you will, that will give us a representation of the physical reality of an experience. That is what we're hoping, that ultimately you could read minds, basically, by seeing the structures that are changed and modified by experience.
It's used in professional articles. I'm blaming Jeff because I learned the word from him.
Yeah, you know, genomics is it's not economics that we're thinking of, but when we think of connectomics or the connectome, we're thinking of genomics and the genome. That is, you map the DNA, that's genomics, and you end up with a genome, which is the full map of all the genes. And there is another map, a structural map of the connections of the brain. That would be the connectome, and you map it by the process of connectomics, the process of making that map.
Okay, so I'm just curious because I haven't heard it until today, but the word is out there and being used in the production.
As a gag.
First appeared in print in 2005.
As a gag, the word connectomics, I start many of my lectures with a dictionary definition of it, with its pronunciation, with all the signs. And at the bottom it says Merriam-Webster's Unabridged Dictionary, 2017. So, you know, it's not, it is a word, but it is, this word is going to be in, the word connectome will be in the dictionary in a year or two, probably.
So the question is, what's the crucial next experiment to demonstrate the importance of neuronal connectomes?
Well, I should tell you that, you know, there is this idea that old dogs don't learn new tricks very well. And there is a connectomic reason for that. It's that their wiring diagram is stuck in a particular form, and new ideas don't come in very well. So you have to kind of wait. I'm not sure anything I can or you can say will convince some of our colleagues that this is a good idea. We have to wait for them to die, and because I have gray hair, it's going to be a race to see who dies first. I think so. We hope is that we will train a new age of scientists who are more open to the idea that one will benefit by more information. You know, a lot of us are afraid of information. I should tell you, in the Human Genome Project, there was a great controversy over whether it should be done or not. The argument was made, what would we do with the gigabytes of data? How could we ever deal with gigabytes of data? In the 1960s and 70s, gigabytes of data seemed so much. No one really knew how we would deal with that. And the same arguments are rearing their ugly head again right now, although it now petabytes.
So the book does describe the experiments that are being done. A lot of attention is being paid to perception, the problem of perception. How do we recognize things that we see? There's very I guess 50-year-old theories about how that actually has to do with the detailed wiring of neurons in the visual system. So there's a lot of work in perception, and we're going to see that over the coming years. I think that's exciting. And then the next problem is that of memory, right? If memories are really stored as patterns of connections, can we somehow read out memories from connectomes? And so in the book, I propose this example of birdsong. Birdsong is an example of a memorized sequence. Birds actually learn how to sing. They have some instinctive ability, but they also learn. And the question is, where is that stored? So scientific experiments are described that you can, but it would take a longer talk. It would take a longer talk. So your genome you can actually get from a skin cell, for example, but the only way we know of finding a connectome now is from a dead brain, because in order to get the high spatial resolution we need, we have to slice it up into these incredibly thin slices. So there's a fancy, Jeff in his lab has the most expensive deli slicer you've ever seen. All right? And there was a New York Times article on Jeff which said, this man likes his brains very, very thin. Okay, so you get the idea. And then he has a fancy microscope. Anyway. Yeah, the connectome is not the activity. So the analogy I use is the stream versus the stream bed. The activity is like the water of the stream. The connectome, the connections are like the stream bed. They determine the course of the water. But also, the metaphor is better because the water also slowly sculpts the stream bed. That's how activity leaves a trace. Okay? Well, I think one big message I want to communicate is that we need basic research on neuroscience. So sometimes people think, oh, we'll just develop some drugs, we'll develop some therapies, and we'll have it licked. But we understand so little about how the brain normally functions. How can we have a chance of understanding how it malfunctions, right?
I mean, just a classic example of this is, imagine a car mechanic trying to fix a broken car. You come in, you say your car doesn't work, but the car mechanic has no idea how a car works. And that is how we are with mental illness. You know, there's no doubt something is not right, but there's no physical trace of what's wrong. So the fact that we have any therapies at all is quite remarkable. Most of them have come about by accident. But for many diseases, the autism spectrum disorders, as far as I know, there's no medicines. There's nothing really that is very effective at all because we really don't know exactly what's wrong.
So applications are important, but basic research is also, there's just no way. There's no way to move forward without it.
Yeah, I mean, this is what I study, basically, is why humans come into the world as the most helpless of all creatures, why it takes us a year to walk, much longer than any other animal, why it takes 18 years to leave the nest, if you're lucky. I'm hoping that's the truth. We'll see. But,
He's an empty nester now.
Yeah, just recently. Something is clearly interesting about human brains. We come into the world knowing much less about the world than an insect. Insects don't have to go to school to learn how to hunt or fly. They know all that stuff. We have to learn even how to the language we think with. We have to learn how to think, to use words. We don't have any of that built in. So what is happening to our connectome during development is, I think, an essential ingredient to being human. That's how we go from being this protoplasmic mass that can't even turn over when we're born to these magnificent creatures that are each of us now as adults. And I'll just tell you one surprising result here, and that is when you look at little pieces of the connectome in babies, when they can't do anything, they are much, much, much more complicated than the wiring diagram you end up with. That there's a massive amount of branches that disappear. And what you end up is a small percentage of what you could have been had your experiences been different. And that's why it's hard to teach old dogs new tricks, or why my children call me a dinosaur, because I'm unable to Twitter or whatever the verb is. With life, life is almost always fighting entropy in an interesting way, that we generate a lot of heat in the world, but make ourselves more and more structured. So I don't know whether you can think of us as a box of chemicals that are just reacting. We clearly have a lot of things going on to prevent us from becoming random. We are becoming fixed in our ways and highly predictable. By the time we're old, we're pretty predictable creatures. We have much more entropy and weirdness as children when we can think all these weird thoughts, and you can't leave a baby alone. You can leave an adult dog alone, but you can't leave a baby alone in a house.
So the question is about neurofeedback, which is a relatively new method of personal change, right? So we all know that we can use feedback from a person to actually try to affect change, but some people have therapies which involve looking at your own brain activity in a brain scanner and using that trying to actually use that information to learn how to behave differently. I think it's intriguing. I don't know that there is really solid evidence that it's effective. I mean, there are studies which show some mild efficacy, but I think it's still controversial. But in principle, no reason why it shouldn't. Well, the man Ken Hayworth is not here. Ken actually works in Jeff's lab, and he invented the deli slicer. Ken has founded something called the Brain Preservation Foundation, and he's offered a $100,000 prize to anyone who can successfully demonstrate the preservation of the connectome of a large brain, say a pig's brain or larger. So there's a cash prize. I don't think anyone has demonstrated that yet. But there's some people who want to enter his competition.
I think one view is that if you want to model a brain, if you assume that the connections are somewhat stochastic and random, you can get pretty far, and it's a lot easier to model than if you say that every single connection has some meaning. So there's an argument about how much noise there is in the system. How much detail do we really need? They say, are you crazy? You really want to get every wire? I mean, that's ridiculous. You don't need all that. There's some huge amount of noise. You can understand a lot by doing much more crude things. And there are some of us who say, well, maybe the detail is really where all the information is stored. It's not that this part of the brain is connected to another part of a brain, or that this part of the brain connects to this layer in this part of the cerebral cortex. It's exactly which connections are there. And that's where the argument is. Most people are not connectionists at the level we are connectionists, yes. And they think it will cost a lot of money, and that money could be going into things that they're doing. It's a perfectly reasonable argument.
Okay, so what I would say, I haven't read those papers, but there's many papers like that. There's many papers like that. What they usually show is a statistical correlation between some brain property and that mental property. And that correlation is usually weak. So you can't, for example, there is a correlation between brain size and IQ. There actually is a correlation. But you cannot predict someone's IQ based on their brain size. And so, same thing with autism. Autism is correlated with a larger than average brain and head as being a child. But if you took every child with a large head and said they're autistic, that would be a worthless kind of prediction. So these are all tantalizing hints. These correlations are tantalizing hints. But I think until they get to a stronger level and a more predictive level, it's not going to tell us enough. So there are steps in the right direction, but we're arguing basically that we need finer measures in order to get that stronger correlation. Okay, so that's perfect the question to conclude on. So Jeff talked about how he's producing this deluge of data. And in fact, what I do every day in my lab is to develop methods for analyzing that data. And so we have all these images, but these images are too much for any person to look at. So we have worked for quite a few years on making artificial intelligence, making computers that can see the paths that the branches of neurons take inside the brain and see synapses automatically. But it turns out that, as you all know, although robots see really well in science fiction movies, they still don't see very well in reality. So computers are still not as good as humans at seeing. So we also need people. We need people to interact with the machine and correct the errors of the machine. And this is really where our research is leading to. We've just constructed a website. So some of the students in the class that helped us construct it, they're actually here. I see Elizabeth and Emilio. It's called iwire.org, so EYE, and then wire.org. And what it means is that any person in the comfort of their own living room can actually come to this website and help us map the brain's connections by analyzing these images and working with the AI. So you could think about the brain as an incredible jungle. So I say we have to unite to explore the jungle of the mind because neurons look like trees. And so I'd like to just conclude with actually the opening of the book and leave you with that thought. No road, no trail can penetrate this forest. The long and delicate branches of its trees lie everywhere, choking space with their exuberant growth. No sunbeam can fly a path tortuous enough to navigate the narrow spaces between these entangled branches. All the trees of this dark forest grew from 100 billion seeds planted together. And all in one day, every tree is destined to die. This forest is majestic, but also comic, and even tragic. It is all of these things. Indeed, sometimes I think it is everything. Every novel and every symphony, every cruel murder and every act of mercy, every love affair and every quarrel, every joke and every sorrow. All these things come from the forest. You might be surprised to hear that it fits in a container less than 1 foot in diameter, and that there are 7 billion on this earth. You happen to be the caretaker of one, the forest that lives inside your skull. The trees of which I speak are those special cells called neurons. The mission of neuroscience is to explore their enchanted branches, to tame the jungle of the mind. Thank you all for coming.